Deoxyribonucleic acid, more commonly known as DNA, is the molecule that acts as a blueprint for all cellular life, holding the instructions of how to carry out all processes that keep living things, well, alive. We know this now, but there was a time not too long ago when we didn't know what DNA did. However, after a successful experiment, we made this exciting discovery. Today I will be discussing the Waring blender experiment, performed in 1952 by researchers Alfred Hershey and Martha Chase, which proved that genetic material is made from DNA. Part 1. Before the experiment. What did scientists know about our genetics before this experiment? Well, we knew that DNA existed, and we had a pretty good idea of its structure. We knew that DNA is made up of repeating subunits called nucleotides. However, we didn't really have a good understanding of what DNA did. We also had a relatively good understanding of genetic material. We knew there was something in our cells that were passed to us through our parents that gave us unique characteristics. However, we didn't know what genes were made of. There were many scientists who believed that genes were made of proteins and that DNA maintains cell structure. There are many different configurations of proteins, whereas DNA nucleotide structures are very simple. Therefore, the variability of genetic material was thought to be due to the variability of protein configuration. Hershey and Chase were not the first ones to set out on a mission to prove that DNA had a role in genetic material. In 1944, researchers at the Rockefeller Institute discovered that DNA facilitated a process called bacterial transformation. What is bacterial transformation? Bacterial transformation is a phenomenon where bacteria cells share genetic material with other bacteria cells that change the characteristics of the bacteria. Through their experiment, they realized that DNA was what was shared between the bacterial cells. Therefore, DNA is what changed the characteristics. Even so, there were still many scientists who believed that this did not prove that genes were composed of DNA, and they still believed genetic material was made of proteins. Hershey and Chase's experiment set out to prove these scientists wrong. Part 2. The experiment. Alfred Hershey was a virus researcher at the Carnegie Institute of Washington in New York. Along with his associate Martha Chase, they started planning a way to model the movement of genetic material in order to determine if it was made of protein or DNA. They came up with a fascinating experiment called the Waring blender experiment. In order to understand this, we first need to understand two concepts: bacteriophages and radioisotopes. What are bacteriophages? Bacteriophages are a type of virus. Viruses, as we know, are infectious organisms that infect and attack cells. Bacteriophages are special because they only infect bacteria cells. They are made up of an incredibly simple structure: DNA, an outer protein coat, and a tail. This makes bacteriophages perfect for the experiment as they only consist of protein and DNA, which are the materials we are trying to isolate. When bacteriophages infect bacteria cells, the tail attaches to the outside of the cell, and then something, we don't know what yet, is injected into the cell. After the bacteriophage infects cells, it becomes a bacteriophage ghost that stays attached to the cell, although it's just empty. The bacteriophage causes the cell to reproduce many other bacteriophages, and then lyse, which is when the membrane essentially explodes, killing a cell. When the cell is lysed, the produced bacteriophages then move on to other bacteria cells. What are radioisotopes? Radioisotopes are a type of isotope. Isotopes are different versions of one element that have the same number of protons but a different number of neutrons. Regular isotopes are stable, but radioisotopes are isotopes of an element that are unstable, meaning they randomly break down, and as they break down, they emit radiation. This radiation can be tracked by scientists. This is called radiolabeling. Now let's get back into the experiment. This was their concept. They took two samples of bacteriophages. The first sample was infused with a radioisotope that tracked the movement of the DNA, and the other sample was infused with a radioisotope that tracked the movement of the protein. They would introduce the bacteriophages to bacteria cells and allow them to infect. Then, they would agitate the samples with a special blender in order to remove the bacteriophage ghosts from the bacterial cells they infected. After, they would centrifuge both samples. What is centrifuging? Centrifuging is when you spin a sample so fast that the different parts of the sample with different densities separate. In this case, they wanted to centrifuge the samples such that they separated into the bacterial cells that had been infected and a liquid medium that contained all of the separated bacteriophage ghosts. Then, they tracked where the radioisotopes were in both samples. The sample that had the radioisotope within the bacterial cell section and not in the liquid medium section was the sample that the genetic material is composed of, because that sample tracked the movement of either protein or DNA from the bacteriophage into the bacteria cell. How did they choose the bacteriophages, bacteria cells, and radioisotopes they used in the experiment? They chose a bacteriophage called bacteriophage T2 that infects E. coli cells. As for the radioisotopes, well, sulfur is found in protein but not in DNA. So for the sample that would track the protein movement, they chose a radioisotope of sulfur called sulfur-35. Similarly, phosphorus is found in DNA but not in protein. So for the sample that would track DNA movement, they chose a radioisotope of phosphorus called phosphorus-32. So, one fateful day, they did the experiment. What happened? Well, for the sample of bacteriophages that was tracking the proteins, they were introduced, they infected the E. coli cells, were agitated to split off the bacteriophage ghosts, and then centrifuged to separate the ghosts and the bacterial cells. They were then tested to track where the sulfur-35 radioisotopes were. They discovered that the radioisotopes were with the bacteriophage ghosts. This means that the proteins had not been injected into the bacterial cells, and that genetic material was not made of protein. For the sample of bacteriophages that tracked the DNA, the same process was repeated and then tested for phosphorus-32. The results? The radioisotopes were with the bacteria cells. This means that DNA was transferred from the bacteriophages to the bacterial cells, and that genetic material is made of DNA. Part 3. Impacts. What did this experiment teach us? Well, it taught us that genes are made of DNA. DNA is what is passed through generations and what carries the instructions for all life processes. This was an incredible discovery in genetics research. Without the results of this experiment, we wouldn't have the foundations to find other amazing discoveries that led the way to learning about technologies such as genetic testing and gene therapy. This experiment opened many doors for scientists to understand more about how we work. Thanks for watching. Hopefully this video helped you gather a deeper understanding to the Hershey-Chase experiment and how it impacted DNA study forever.